So again, my name is Matthew Hess. I'm with BennuBio. BennuBio is a biotech startup here in Albuquerque, and I've got some slides just to give you a little sense of what we're doing and where we're heading. So our instrument is, our product is called the Velocyt, and it is a flow cytometer, which means it is used for studying cells or other particles in suspension. And what differentiates it, we call it parallel stream acoustic focusing flow cytometry. And it's a leap forward from other flow cytometers in several ways. So there may be people on the call who are familiar with flow cytometry. There may be people who have never heard of it. The key points are that a typical flow cytometer tops out at things that are about 40 microns in diameter. Generously, most of them can't go that high. We are able to easily see things that are 500 microns, and we think we can go bigger, maybe even up to 1 millimeter. So we're able to do at least 10 times the size, which opens up a lot of avenues. A typical sheath-based flow cytometer has a sample aspiration rate that, again, generously tops out at around 120 microliters a minute, and we can do 10 milliliters a minute. So it's about 100 times more throughput by volume. Typical flow cytometers, just by the nature of how the hydrodynamic focusing works, create kind of a high pressure environment for the samples, which can be stressful. We have a very low pressure system, so it is more gentle on the samples. Typical flow cytometers are prone to clogging. It's just the nature of the beast. You've got 150 micron capillary, stuff accumulates and they clog up. Our machine is virtually uncloggable. And in a typical flow cytometer, everything you run through it ends up in a waste bottle with a bunch of diluted, by 90% sheath, and sometimes with bleach and other things. So it's what's called a terminal assay. You basically run your samples and that's it. With our system, the sample is fully recovered, which allows you to run the same sample multiple times, doing what are called kinetic assays, or to use it upstream of other analytical tools. So we do bigger things, we do them faster, we do them more gently, we tend not to clog, and we give you your sample back. Now, having said all that, it's important to note that flow cytometry is the gold standard for cellular analysis. It gives you high throughput, multiplexing capability. I've worked on flow cytometry instruments and for companies for over a decade. I love flow cytometry. So this is not a knock on existing flow cytometers, many of which are amazing. It's just that we've kind of built a new design which allows you to apply all these amazing flow cytometry techniques to a whole world of assays that you couldn't before. People have wanted to, but they couldn't because of these limitations. So we're just expanding the horizons. Just a little more on the technical side. The particles pass through that flow cell, we hit them with lasers, and then we collect the fluorescently emitted or scattered light, pass it through mirrors and filters, and then capture it on a high-speed CMOS camera. Again, doesn't really matter from a perspective of why is the technology important, but there may be some technical folks on the call who are interested to know that's how our optics work. And we take that image data and we turn it into standard flow cytometry metrics. Again, it's called FCS data is the format. And there's a whole set of widely available software tools built to analyze flow data. So it's really easy to analyze the raw data that comes out of the machine. We do mention, though, that in addition to that basic flow data, because we have the camera going on and we're getting multiple pixels per event, we're actually able to do some more 3D analysis that's not available on traditional flow cytometers, which helps us measure things like drug penetration, uptake, cell proliferation, and necrosis within these larger 3D tissue models that I'm going to talk about. So that's the Velocyt Concept. That's our instrument. That's what's cool about it. The question then, of course, is why should you care? Right? Who needs this? Does it solve real world problems? Because people don't buy things because they're nifty. People buy things because they solve their problems, because they need them. So the next part of my presentation is reviewing some of our applications. So one of our most interesting applications has to do with spheroids. As you know, people, this stat gets bandied about, but up to 90% of new medicines fail in clinical trials. They looked good in the lab, they get out into the field and they don't work. So why is this? A lot of researchers think it really has to do with the fact that a lot of that early in vitro testing is done on single cell models. You grow some cells, you treat them, you test them, and you learn about your treatment. And of course, organs, tissues, tumors are multicellular. So a lot of emerging research, particularly in the area of cancer and solid tumors, uses cell clusters called spheroids or organoids. And the idea is that these clusters are a community of cells where the cells are communicating and functioning more like the actual tumor or tissue, and so therefore they provide a better model. The problem is they're big, from like 50 to 250 microns, and they're often a little bit fragile. So you can't run them through a traditional flow cytometer. People have tried. What you do is you have to disaggregate them down to single cells. You basically have to break them, and you lose a lot of information in that process. So the Velocyt is the only instrument out there capable of high throughput multiplexed analysis of spheroids in suspension. So we feel like almost anybody out there doing cancer research, particularly on solid tumors, has a need for this technology. Another possible application, and I will say, like with the spheroids, we actually have 2 active collaborators in our pilot program who are doing this. So this isn't theory, right? This is real. We're starting, we run these in-house ourselves, and we're starting with customer samples this month. Cardiomyocytes are your heart muscle cells, and they're about 120 microns long. They're sort of brick-shaped, and so they're too big for traditional flow cytometers. And what that means is that people are basically stuck with microscopy as a way to study them. And there's a lot of information you can do, you can glean through microscopy, but you don't have that multiplexed power of flow cytometry. So interrogating, running a ton of them, like tens of thousands through, and asking questions about their size, geometry, and in particular their cell surface biomarkers really isn't something people have been able to do at all. And what we're working with a collaborator at a university in California who wants to take various heart tissues from genetically modified mice and also from human transplant patients where there's tissue that it's been removed because they received either a partial or complete transplant, disaggregate them down and then study the cardiomyocytes, and for the first time get real population level information about these really important cells. And it can't be done on any other instrument. Another application, induced pluripotent stem cells. These have gotten a lot of press. People are aware of stem cells, but basically iPSCs can propagate indefinitely and give rise to any other cell in the human body. So they have huge potential in the field of regenerative medicine. And
Matthew, you have a minute left.
Okay. They tend to aggregate naturally, and so they're very difficult to study, but our platform will let you do that. Immune modulating T cells, CAR-T and K-CAR is another area where we've got active collaborations. These are cancer therapies. CTCs are rare events in human blood, also very difficult to study because blood is a difficult sample. It requires lots of sample prep that we can get around. Microembolii are little clots that create lots of health problems, and even things like algae, which have applications with water quality and environmental toxicology, are all targets for our platform. All of this invented basically by our founder and CEO, Steve, who's at UNM. We've got an amazing team of people here in Albuquerque working on this. And basically our plan is to focus on commercializing the instrument. They cost about $100,000 each. And we're planning a 2-laser 8-channel model later this year. In addition, we expect revenue from consumables like cleaning kits, reagent kits, and service like IQ, OQ, PM, 21 CFR Part 11 support. So that's the business plan. We've been venture funded. We got $7 million in venture funding so far. We're actually looking for some more. And that's the summary of BennuBio.
All right, thank you. Andrew, Ben, do you have questions?
Absolutely. Matthew, thank you for this great presentation. How much more money are you looking for, and what are the uses of those funds?
So we have $2 million open tranche in Series B. So the terms are done. It's really just kind of a shoe-in for somebody who's looking for that, and it's funding the commercialization efforts, right? We have a business plan, and we're really strong in terms of science and engineering. This is actually the best R&D group I've ever worked with, but we need to hire production people. We're in the process of all kinds of operational things, putting in an inventory system. We need to hire a production manager, production technicians, field application scientists, field service engineers, salespeople. It's basically the whole operational and sales side to now actually commercialize.
So will this be direct sales?
Yes.
Okay.
Eventually, we'll need a distribution channel for like Singapore and China and Japan. It's very difficult to access those markets directly, but we're planning direct sales in North America, UK, and Europe. And that's one of the reasons they hired me. I actually have done this before and have good connections in all those places. I've got folks asking me if I can hire them yet in the UK and Europe. I'm like, okay, guys, you got to wait a year, but we'll be there soon.
Very good.
Great. Matthew, this is Andrew Moore from Bluestone Venture Partners. Thank you again for sharing that with us. One of the questions I had, being able to analyze, particles at high throughput is good, but depending on the use case and, for example, cancer researchers, they need to identify subpopulations and pull out the rest of the particles and sort through them. So are you guys able to pull out and sort any of the particles you go through, or is it just analysis and high speed, high throughput analysis?
So we basically have the plans for the sorter. There are a couple of really important like add-ons and upgrades. One is more lasers and channels. One is automatic sample loading, like a carousel with Eppendorf tubes or 96-well plate format. And then the killer app, the one you've mentioned, is the sorter. So that is definitely in the plan. And in fact, we're working to get another SBIR grant to help fund that. What's important is we have to solidify the base platform first, because the sorter relies on the core technology being bulletproof. Okay. So the way I'm looking at it as an operational guy is I want us to focus on, let's get Velocyt nailed down. Let's sell a bunch and refine that and make sure that we're really solid on our core analysis technology, and then we can add sorting onto it. But it is actually actively being worked on. It's just not something we're really promoting or leading with. I'm very cautious about, selling things that aren't quite ready, right? So the answer is yes, we need a sorter. We will have a sorter, but not yet.
Got it. And would that be a Series C round later on that you guys would raise for, or would any of the use of funds be for the sorter?
I don't actually think we're going to need to raise Series C for the sorter. I think when the time comes, we will look at Series C, but it'll be more for something like strategic acquisition to partner with reagent companies to really build our portfolio, or some of these things you can probably tell have potentially clinical applications. So for example, one of the rare events applications is running human blood and monitoring minimal residual disease, and also monitoring persistence of CAR T cells. And we will be running patient samples with those collaborators. And if it looks like there's an actual FDA-approved clinical application, then you have to go to round C for a fairly substantial chunk of money to take you over that threshold. So we don't want to do round C as an incremental thing. We want to do it as, okay, now we're going to go after something really big. And I think between the NIH funding, SBIR, and the revenues we'll generate off the base instrument, we'll be able to self-fund the sorter.
Got it, thanks.
Great questions. Thank you. I'm pretty sure Bluestone's kind of familiar with our portfolio. We've had some conversations. Right?
We have. I guess I have another question, I guess, unless Ben has one. Just kind of understanding how you guys will be able to get, researchers or clinicians to adopt your technology. There are a lot of competitors out there and a lot of very highly sophisticated machines, and a number of smaller companies that have never really been able to scale rapidly. So how do you guys plan on differentiating yourselves well enough to be able to hit that really big growth curve?
That's a great question. And it's one that's near and dear to my heart. It's actually why they hired me, because this is the thing that I focus on. I was at Velocyt for 11 years, which was another Albuquerque-based flow company. And a lot of what I was senior software engineer to start with, but later on, I really focused on the customer side and instrument adoption and technology adoption. So the first thing to note is we really have no direct competitors. If somebody wants a full-featured flow cytometer, multispectral, they just go buy a Cytec, which is the state of the art. Or they go with BD if they want tried and true, right? And yes, there's 100 competitors there. You can get very affordable things like in ASEA, Novocyte, fantastic technology, but it's not a competitor because they can't do all these things that I mentioned at the outset. It's complementary, really. So we're not going to try to go head to head with those folks, right? We would lose. It's not going to work. We're going to solve new problems. So what our strategy is fairly straightforward. And what we're doing now is we've launched a Velocyt pilot program, and we have 3 collaborators, and I'm actually able to share their names because they've signed the doc. So Baylor College of Medicine, Precision Biosciences, and TD2. Baylor is doing amazing cutting-edge cancer research, and we've got a pilot program to do an apoptosis spheroid assay with them. Precision Biosciences has a CAR T-cell product, and they want to monitor persistence post-treatment. And TD2 is a CRO, which is kind of a full-service CRO helping all the big drug makers bring their products to market. And we're going to be doing essentially small molecule screening using 3D cell models for them. So the plan is work with these collaborators, generate data, generate some publications. This is what we really need, is peer-reviewed, publications showing, hey, this technology can do this thing. And okay, it's probably too simple to say, then we sit back and take orders. But like if you look at Accuray, they kind of did.
Matthew, we're going to have to wrap up here.
Okay,
so,
one sentence. Once Accuray proved it, they really did take orders. That prospecting and selling became quite easy. So that's the goal. Prove it, publish it, and then I think prospecting and marketing becomes straightforward.
Thank you.
Great. Thank you so much, Matthew. Really appreciate your presentation.